Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really glad you made time during this nice month of December to dial in here today. And we will be talking about drugging NLRP3 with Angela MacKay today. Good afternoon, Angela.
Hi, Dennis. Good afternoon.
How are you?
I'm good, thanks. It's evening in Basel. I'm in Basel, so yeah.
Yeah, and it's morning here in San Francisco. And now, without further ado, I want to formally introduce our guest, Angela MacKay. So Angela's love affair with chemistry started really early whilst she was at Scotland in school. And pursuing an undergrad in chemistry at the University of Edinburgh was somewhat of a no-brainer for her. She then moved to the University of Cambridge in the UK, where she earned her PhD in total synthesis of natural products under the mentorship of Ian Patterson. Exchanging one Cambridge for another, Angela pursued a postdoctoral research position at MIT with Professor Stephen Buchwald. And in 2004, she joined IRBM Merck research labs in Pomezia, Italy, where she worked on antiviral targets. Then in 2009, Angela joined the Novartis Institutes for Biomedical Research in Basel. And here, where she's still working today, she works across different disease areas with a particular focus on immunology and neuroscience. And she currently leads a group that's focused on neuroscience, aging, and regeneration. Angela, such an honor to have you here today. And Angela and I know each other from our time back at Novartis. So welcome again. And yeah, happy to be with you today.
Thanks.
Awesome. And yeah, in our good old Drug Hunter tradition, we like to start with some warm-up questions. So I wanted to ask Angela, what inspired you to start a career in drug discovery?
So I think, as you said in my little intro, I've always been interested in chemistry. I actually got interested in chemistry because my mum's a pharmacist. And so that kind of sparked my interest in chemistry from a very early age. And when I started to learn about chemistry at school, organic chemistry, it's like cooking with a perfect mix of cooking and witchcraft. So that was always something for me that was really really cool. And then the fact that we can use our organic chemistry to manipulate and impact biology. It's not the biology itself that I like, it's the fact that we can use our chemistry to manipulate it. And the fact that we can do that with living systems and to improve human health, I think that's really, the most inspiring thing. So I am doing my dream job. This is what I always wanted to do. So yeah, for me, it's just always been the thing.
Amazing. Yeah, I think many of us in the audience, including myself, share this exact passion with you. Yeah, with that, what's your favorite molecule and why?
So this is an easy one. It's calicheamycin, the anti-tumor, the indie anti-tumor antibiotic. And the reason is, so it was my first, when I was at Edinburgh University, I was asked to write an essay. It's not very often that as chemists we're asked to write an essay, but we were given a list of topics. And one of them was to write an essay about the indie antibiotics and to explore that. And so, I looked up these papers and I thought, wow, how cool are these? The structure is amazing. They're gorgeous. And, the function, the rearrangement, the benzyme, the fact that they slice through DNA, I thought that is absolutely amazing. And so that really, the reason I pick it as my favorite is because it's the moment that I realized what I wanted to do for my PhD. I wanted to do natural product synthesis. And so from there, it was fairly easy to work out who's doing natural product synthesis in the UK. And then going to work with Ian Patterson, these huge polyketides. Polyketides are the most beautiful molecules. And so for me, that was the calicheamycin story was sort of the jump off point. So.
Amazing. Yeah, aldol reaction may be your favorite reaction then.
The aldol reaction is absolutely my favorite reaction. with boron, preferably.
Great. Sounds good. What are you most excited about in drug discovery today?
So I was trying to think of something that wasn't AI. I guess that everybody every week answers that. And I think it is the thing that's going to really impact our industry most. but I mean, I'm not really a computer person. I'm very much an end user. And so for me, the reason that I think it's the most exciting is because I see drug discovery as like, I like to do jigsaws when I was young. So it's like a big puzzle where you don't know what the picture is. It's like somebody threw away the box before you started, right? You don't know what the picture that you're trying to make exactly is. And so I think, what the AI and machine learning models and physics-based models can do is to clarify that picture faster. And so I think, if we can use these models, I don't think they're going to eliminate us quite yet, but I think, if they can help us to clarify what we're trying to do and to guide us, I think that will be really, really exciting. And I see that already in some of the projects that we're working on.
Yeah, it's always good to see kind of the real world impact where there's a lot of hype typically, right?
Yes, there's a lot of hype, but it is really helpful. And also not just the AI ML models, but the way that we can analyze data and visualize data, I think is also extremely useful. So that's cool. Yeah.
Super cool. Well, if you could go back to the kind of beginning of your career and give yourself some career advice, so maybe some career advice for the younger folks in the audience here today, what would you say to yourself?
So, I mean, when I first started, you put a lot of pressure on yourself. And when you're a PhD student, or mainly a PhD student, maybe less as a postdoc, you define yourself in certain ways. So, I defined myself by whether or not my aldol worked and whether or not you could remember the mechanism of some highly obscure organic reaction, that my extremely intelligent colleagues could always remember. And so that was how you benchmarked yourself. But actually, when you're doing drug discovery, there's really an awful lot of skills that you need to really be successful in drug discovery, not just organic chemistry at the bench. Obviously, that's very important. But you need to be good at other things, data analysis, leading people, leading a team, knowing when to take risks, knowing how to evolve a strategy for your project. And so I think if I'd known 20 years ago that, it wasn't just about whether or not my aldol worked, but there were a whole load of other different skills that you also had to excel in, I think that would have helped, because you don't have to be good at everything. That's the point. And so sometimes if you're in a peer group where, maybe you feel that, that person's better at this and that person's better at that, you will find your thing that you excel in and where you are the best at. And so to keep that in mind, it's just to work out what it is.
Amazing. Yeah. That's, I think, very good advice. Yeah, thank you. No, you ought to be like, often like people feel like imposter syndrome, I feel.
Yeah, it's a bit like that, yeah.
Yeah. What do you most like about Drug Hunter?
So I think that what Drug Hunter does is what I really like are the articles. They're kind of like review articles that drop into my mailbox, about a particular topic. It could be something medicinal chemistry about like a target. Maybe it's something that I've never worked on. And so it's something that I've never really explored. And it gives you like a short overview, something that's highly readable. I can read it on the train. And it gives me like a window into that area of science. And maybe that's all I need to create a basis. And then, or maybe I dig a bit deeper. I mean, I think this morning I saw something about the FDA designations, and you had a really nice little overview about the FDA designations. And that is something that's super complex to work out as you go through your career. People talk about FDA fast tracks and, waivers, and you think, how does all this work together? And so when I saw that this morning, I thought that's exactly what Drug Hunter does, because you sort of demystify and simplify. And so I think that's really, really powerful for people.
Awesome. Thank you so much, Angela. That's very much appreciated. And that's, in fact, the article of the day. So, yeah, I did want to...
I liked it.
Yeah, that's great. That's amazing. Good to hear. Thanks for that feedback. Yeah, I did want to show people very quickly a snapshot into Drug Hunter. So, yeah, this is the article that Angela was referring to here with the guides to the FDA designations, where we untangle all of this. I also want to encourage people to go here, as Angela pointed out. You can just type in a target, for example, NLRP3, and you see different molecules come up. You can see, different molecules that are related. You can see target reviews and much, much more. Today, I wanted to highlight the structure search, actually. One of the things that I really like about the structure search is that it is very much differentiated from other structure searches in that, for example, you can do single atom structure searches. So, for example, if you wanted to know what are very exciting molecules, drug molecules that contain phosphorus, you can just put in phosphorus, right? And if you would do that in other like databases, you would get really overwhelmed with a lot of things, had to probably comb through millions of hits. And here with Drug Hunter, we really curate it for you. We give you kind of different modern use of phosphorus here. And you can look at Drug Hunter molecules. You can look at FDA-approved drugs. I have that open here in another tab. You can see FDA-approved drugs or clinical candidates. And they're all indexed here with the target when we publish them. And if you wanted to learn more about them, there's also, we typically also have case studies on those molecules. So for example, here, if we wanted to learn more about this ORAC molecule, you can directly click into here and see a full-on case study, learn more about the target, learn more about how this molecule differentiates, learn more about the competitive landscape in this space, and be just much more informed. But it's so difficult to find structure search where you can just use a very much single atom. And here I just tried this with phosphorus, and it gave us a highly focused list that's highly curated, with a lot of relevant structures in here. And with that, I will hand it over to Angela for her talk on targeting NLRP3. DFV890 and beyond. Very excited for this talk, Angela.
Okay, so today I'd like to share with you the story of NLRP3 at Novartis, basically. So the IL-1 beta pathway is one of the most important inflammation pathways in the human body. The pathway is triggered by a variety of different pathogens and danger signals, and each of these danger signals has a different sensor protein. The NLRP3 is the sensor trigger protein for sterile danger signals. Sterile danger signals are things which are self to the body, so not things like viruses or pathogens, but things which are produced by the body but which are disease-causing. When NLRP3 senses these danger signals, it comes together with a variety of proteins to form an inflammasome complex. So these are ASC, which is a scaffolding protein, NEX7, and also caspase-1. And all this inflammasome complex basically does is it allows caspase-1 to mature and perform its protease function. So caspase cleaves the pro-cytokines into mature IL-1 beta and mature IL-18, allows them to leave the cell. And then eventually what happens is the cell undergoes a form of pyroptotic cell death, or whereby the cell basically erupts, spilling its intracellular contents into the extracellular matrix. And these intracellular contents are also sterile danger signals, which cause the inflammation cascade to happen. So the NLRP3 inflammasome has been associated with a very wide variety of different diseases, and I show you some of them here. So I always like to use gout as the perfect example of the NLRP3-driven disease. So gout is caused by monosodium urate crystallizing in your joint, typically your big toe. And what happens when this happens? Your joint becomes red, becomes inflamed, becomes hot, and very painful. And this is a classic immune response driven by the IL-1 beta pathway. So in this case, the monosodium urate crystals in your joint are the thing which are triggering NLRP3. In cardiovascular disease, what happens is that you get plaques of cholesterol setting down in your arteries, and it is these plaques which cause the trigger for the NLRP3-driven inflammation. In neuroinflammation, we think about Alzheimer's disease and Parkinson's disease. And in Alzheimer's disease, again, we have a situation where they have these protein agglomerates. So typically it's A beta, which forms plaques and drives the Alzheimer's disease. And these plaques in the brain stimulate the NLRP3 inflammasome and cause inflammation. And similarly with alpha-syn aggregates in Parkinson's. And so really you can see that there's a whole host of diseases which have a high unmet medical need on this slide. And I think that if we could treat half of these diseases with an NLRP3 inhibitor, we would really be on a road to some transformational therapies for human health. So Novartis has been working on this pathway for a very long time. We do have an antibody which has been marketed, which is called canakinumab, an IL-1 beta antibody for this pathway. We've also got a dual IL-1 beta IL-18 antibody in development. We've also looked at caspase-1, as have most companies. Caspase, the problem is always the chemical matter. It's very challenging. We've looked at NEX7 degraders, and then we've also looked at NLRP3 inhibitors. Why do we want to have more for this pathway than just Ilaris? Well, Ilaris only acts at IL-1 beta. If we are inhibiting this pathway higher up at the level of NLRP3 inhibition, we would selectively, only with sterile danger signals, we would stop the release of IL-1, of IL-18, and also pyroptosis. And so you could have effects which go over and above the effects of canakinumab. However, we do have canakinumab on the market, and we have run lots of clinical trials with it. So is there any data from canakinumab which gives us a hint that we could, in fact, have these therapies could have some promise in all these other diseases that we're talking about? So we ran this CANTOS clinical trial back in the early 2000s, and CANTOS was a huge clinical trial for cardiovascular outcomes. We dosed about 5,000 or 10,000 patients over 5 years with canakinumab, and we followed their medical journey, and we were looking really for people that were having heart attacks, but also all the other comorbidities that they had. And so if you look in the medical literature, you can find articles whereby canakinumab is in fact associated with better atherosclerotic outcomes. These patients also had many other comorbidities. If you've got heart disease, maybe you're a slightly older person and you also have osteoarthritis. And what we found is that people that were on canakinumab actually needed fewer hip and knee replacements than those who were not. So that sounds pretty interesting. There was also some effects on homeopathisis in a specific TET2 driven genetic patient population. But the one that I find really, really cool is that people that were taking canakinumab actually had a lower incidence of lung cancer. So if you were taking canakinumab, you were less likely to get lung cancer by really quite a significant amount. And I think that it's a cancer prevention. Essentially, it's not an easy thing to have a clinical trial for, but I thought that was really an extremely exciting effect. And so here we do see the promise of this target. So that's enough about the target and the pathway. What about the drug discovery? So I'd like to take you back to the 90s to start this. And in the 90s, people knew about the IL-1 beta pathway, but they didn't know anything about NLRP3. They didn't really know how the pathway worked. But they were still interested in the pathway. And Pfizer found this molecule, I presume through phenotypic methods, CP456773. And this molecule was an IL-1 beta inhibitor. They had no idea how it worked, but it inhibited IL-1 beta. And so they took it forward into the clinic. And unfortunately, it was stopped in phase 1 for findings that were of liver toxicity. Time goes on, science develops, we gain more understanding, NLRP3 was discovered, people worked out a bit more about the pathway, and in 2015, it was published that this molecule, which is also called CRID3, specifically inhibits NLRP3-dependent inflammation and not any of the other NLRCs or NLRPs. And so at that point, there was quite a few biotechs started to think, well, could we maybe improve upon CRID3 and how would we do this? So a lot of work started back then in about 2015. And then in 2021, the group at Novartis published an X-ray structure really showing that CRID3 specifically binds to NLRP3, how it binds, and the mechanism of how it inhibits the protein. So back in 2015, a bunch of biotechs, including IFM-Tre, looked at this molecule and said, well, how could we improve it? We don't know what's wrong, why it caused the liver toxicity. Obviously, that didn't show up in animal models, or it wouldn't have progressed to the clinic. But one thing that we can say is that it's got a very high human dose. So 600 milligrams BID is a huge drug burden to give to a person. And so if we could try to lower that drug burden, that might give a better molecule. So how do you lower the drug burden? How do you lower the dose? You increase the potency and you improve the PK. If you do that, you're going to lower your anticipated human dose. And so with that in mind, that was really how a few biotechs approached this problem. So if we look at the molecule, I tend to divide it into 3 parts. So starting with the part on the left-hand side, we have this furan. Furans are kind of known toxicophores. They're things that medicinal chemists don't like. We can worry about them being bioactivated and then going on and causing problems when they're metabolized. The quaternary alcohol as well, maybe that might cause some problems. The alcohol could leave. You could end up with a carbocation. So there's certainly some work that could be done there. The sulfonylurea core, very specific geometry to core of this type, very specific hydrogen bond donor acceptor pattern. I think it's quite difficult to replace, but it would be quite good if you could. In this context of CRID3, it's also rather labile, and anyone that's bought this knows that if you leave a DMSO solution on the bench for a while, it liberates the aniline. And so really replacing the core could also be of interest. And then, if the aniline itself, anilines are known toxicophores, and if you're liberating this in the body, potentially that could also have toxic effects. So, going from left to right across this molecule, there are certainly several things that we could think about to change that might improve the molecule. So what IFM did was that they decided to focus, first of all, on the core of the molecule and try to replace that. A fairly traditional replacement may be an NaCl sulfonamide, but IFM placed the bar much higher and said, well, what about something like a sulfonimidamide? Back in 2014-15, this was really not a very well-known motif. The chemistry to make it, there were methods, but not terribly well established, and certainly not used in medicinal chemistry. And so to go into this chemotype was really a big adventure at the time. So what are the differences between a sulfonylurea and a sulfonimidamide? So here you can see some of the properties and the differences in the structures. So when you move to the sulfonimidamide, you actually you cause or you create a configurationally stable chiral center in the molecule, and so you could have separate enantiomers, which can always be interesting. You no longer have an acidic hydrogen. The urea is obviously an acidic molecule, whereas the sulfonimidamide is neutral. And you also enhance that core stability that I talked about. So you no longer have a core whereby you have a high potential to liberate the amine. Obviously, you can undergo metabolic processes which may do that, but you've really enhanced the stability a significant amount. So how that plays out in the properties, obviously you have biggest differences in maybe the log P and log D profile, whereby you no longer have ionizable centers. So you still have a rather polar molecule, but it's a much more balanced profile across the pH range. And a molecule with a log D of 0.46 at pH 7.4 is really rather polar. And then how that plays out in the properties. So if we look at the permeability as we measure it in this fairly standard MDCK line, MDCK cell line, with the sulfonylurea, you have a rather impermeable compound. 5 in this assay is also not the highest number you would like to see, but it's certainly a big improvement. And so we could potentially see that there would be a benefit to going down this route. So what did IFM do? They started with CRID3 and they made the sulfonimidamide. They put it into their cell assay, and what they saw was that you got almost a tenfold increase in the potency in this assay, which you can imagine was a great day at the office for these guys. And that's a really big potency boost, big success right away. However, it doesn't stop there, because of course I mentioned that you now create a configurationally stable chiral center. And when they separated the two enantiomers, they found that most of or almost all of the activity was found in molecule 2, which is the R enantiomer of the molecule. And you're now down to 13 nanomolar in the cell assay. So that's an almost 20-fold boost in potency just with this very I don't want to call it simple because it was really difficult chemistry, and it's not the first thing that everybody would necessarily think of, but just with a rather small change to the molecule, they got their huge potency boost. So they went on and they said, well, the furan, that would be the next thing that we would like to change. They discovered that they could change the furan to a thiophene, and they got the same potency boost. Then they tried to remove the alcohol. So the tertiary alcohol, this is in the context of a pyrazole. The tertiary alcohol actually also had a large part to play in the potency. And here we have a molecule. The starting point has lost a lot of potency, although when you move it to the sulfonimidamide, at least you still get the potency boost. Finally, they went to some other heterocycles, which would have less concerns of metabolic activation, like different thiazole regioisomers, keeping the tertiary alcohol. And again, they found the same potency boost. And this molecule, molecule 9, was found to be 13 nanomolar in the cellular assay. And then in the whole blood assay, where they have an assay with 90% human whole blood, so really an almost just human whole blood, the compound had an IC50 of 180 nanomolar. So this is really a huge boost versus the starting point. The starting point was 760 nanomolar in whole blood. So really you have now a molecule which is much more potent. It's stable chemically, and it's a much better point to move on from. This is actually the molecule that they took forward and that became known as DFV890. So they looked at the potency, but then what about the other properties of DFV? So if you want to improve the potency, you also need to keep the PK at least as good, if not improve it as well, in order to have an improved human dose. And so here you can see the PK profile of the molecule across the 3 species in comparison to DFV. So you can see that the clearance in each case is improved. The half-life is also improved, and the volume of distribution remains very low. And then the oral absorption, you have an improvement there, or in the case of cynomolgus monkeys, it's at least still in the excellent range. And so in this way, IFM-Tre had created a molecule where they had fulfilled their goals of really improvements in potency and improvements in PK, which would surely improve the anticipated human dose. Here we show that the molecule works in vivo. So this is the PK/PD model for NLRP3. This is our workhorse model, which everybody across the industry uses. There are various different flavors of it, but essentially you dose the compound before you stimulate the inflammasome, because once you stimulate, you can't stop the train, if you like. So you have to have compound on board first. Then at a certain point afterwards, you dose with a trigger. In this case, they dosed with LPS, so lipopolysaccharide. And then about 5 hours later, you can terminate the study. You can sacrifice the animals. You can look at the PK and then also wash out the peritoneum. So the LPS is dosed into the peritoneum of the animals. You can wash out the peritoneum and look for the IL-1 beta response. And so here you can see that with no DFV, this is the placebo group, you have a nice response with IL-1 beta, and that is inhibited in a dose-dependent manner as you dose higher and increasing doses of DFV890. This is also the IL-18. So in this version of the model, you can see the IL-18 coming up as well, and that's also inhibited in a dose-dependent manner up to 10 milligrams per kilogram. And this is just all the experiments that were run, put together, all the individual animals, and you can see a nice sigmoid response curve in line with the increasing concentration of DFV in the blood. So you can tie the PK to the cellular, the PD to the cellular assay as well. So here I show that the IC50 in this experiment, the free IC50 is 15 nanomolar, and in a mouse BDMC, so bone marrow derived macrophage assay, the free IC50 is also exactly 15 nanomolar. It's almost too good to be true. The IC90, where we see complete response, is calculated to be 133 nanomolar in this experiment, and about 60 in the bone marrow derived macrophage assay. Obviously, when you go up to the top of the curve, it's a little bit more to get precision, but I think within twofold is really good, and it shows that we need IC90 at trough to get complete inhibition of the pathway. So DFV has the sort of preclinical profile that you would expect of a compound that we take forward into the clinic. Importantly, in GLP tox studies, It had appropriate safety windows to support the advance into the clinic. And using all the data that had been put together, it was calculated that this compound would have an anticipated human dose of between 30 and 60 milligrams QD. So if we think about the starting point where Pfizer were dosing 600 mg BID, I think you would agree that IFM really fulfilled what they set out to do. And we now have a nice low dose, potentially QD compound in the shape of DFV890. So it was at this point that Novartis decided to in-license this compound, and we took over the development. Now, one of the first things that we did at this point in our story as well, we'd managed to achieve that we could routinely get crystal structures of compounds, and so we put DFV into our crystal structure workflow, and we got a structure. Here you can see how it binds and how it closes, how it inhibits the protein. So this is the NACHT domain of NLRP3, and the NACHT domain, when it's activated, it opens out. And these molecules, they bind to the 4 domains in the center of the 4 domains, and they keep the protein glued shut. So you can see the 4 domains are colored in different colors, and the molecule sits at the center of that and keeps this protein glued shut, and it can't activate. Here you see the individual interactions. More specifically, in the back pocket, we have a very lipophilic environment where we put this tricycle. And then the core of the molecule really sort of holds the two sides, or the 2 domains above and below in a closed conformation with the alanine 228 and alanine 227 interaction. This also explains why you got the boost in potency, because this nitrogen can now form a hydrogen bond to the NH backbone of alanine 227, which would not be possible if this was a standard urea. And the carbonyl interacts with this key arginine residue on the other side. And then we have the glutamate, which also interacts with the alcohol. And so you can really see why we needed each of the motifs in order to attain an excellent potency with this molecule. So the molecule did go into the clinic, and then we have the results of the phase 1 study. So this is the multiple ascending dose. We dosed different dosing regimes, so QD and BID, different forms, so crystalline solid and also spray-dried suspension. And what we saw was that we had a nice dose proportional human PK across the dose range, and there wasn't too much difference between the different formulations either. What we also did was that we took blood from the patients which had been treated with the drug, and then we stimulated the ex vivo blood to stimulate the production of IL-1 beta. And you can see here that we did, in fact, get a nice stimulation in the placebo group. But when we had the treatment with IL-1 beta, it diminished. And with either 30 mg of the crystalline suspension or 100 mg of the spray-dried, the spray-dried material, you got essentially complete inhibition of IL-1 beta in these treated people. So this is a whole blood assay in a real patient. And this is just all the data that was collected, plotted in a graph, just to show that you had a nice dose response across the concentration range. So this work has been published by my colleague Eva in Translational Clinical Science. So that's really the story of DFV890. It's a very interesting core morph installing a sulfonimidamide, which provided significant improvements in both the potency and PK of the molecule versus the starting point. DFV is a potent and selective inhibitor of NLRP3. It went forward into the clinic. It showed very good PK and tolerability, and a low twice daily dosing will probably be the optimal dosing regime to cover IC90 at trough in the studies which are going on at the moment, which are in a variety of different diseases, covering a little bit the human being that I showed you at the beginning. So that's the story of DFV, which, as I say, we in-licensed from IFM-Tre. But what was Novartis doing at the same time? So we were also working on NLRP3 at the same time as these activities were going on, and we knew that there were people working on CRID3, and we thought, well, we could do the same, but we could also try to do something different and really try to expand the world of NLRP3 inhibition and find something that's structurally different. If everybody's working on the same thing and we find there's an inherent problem and they can't go over the liver tox, that's not going to be helpful. So let's try to add some more strings to our bow and try to find some diverse chemical matter. So we did that through an imaging-based pyroptosis protection, HTS. To do that, we labeled the ASC protein in the cells with GFP, with green fluorescent protein, and THP1 cells. And we screened the entire Novartis deck against these cells, stimulating NLRP3 with nigericin. And what we were looking for was cells which were alive. We were looking for nice, viable cells that didn't have any problems. And that meant that all compounds which didn't inhibit the inflammasome or which caused other cellular death were removed immediately from the results of the screen, which is sometimes a problem in phenotypic screens where you get pains and such like. So that left us with about 8,000 compounds, and then we started to slice up the pathway. So we were looking for compounds that inhibited NLRP3 or above. The cells that you see down here, you can see green dots. Now, the green dots are the inflammasomes. So that means that the ASC has come together and has formed the inflammasome complex. So that means that you have inhibited below the level of inflammasome formation, could be a caspase inhibitor. And so we wanted to see nice, healthy cells where there were no inflammasomes formed. And then we put the compounds that had shown the effect there, and we put it into our counter screen to look at TNF alpha secretion, so that's upstream of NLRP3, and we ended up with about 140 compounds. We clustered them, we looked across multiple stimuli, because obviously if this is really targeting NLRP3, it should work across a whole range of stimuli, and we did some other basic profiling, and we ended up with about 10 hit compounds or series. So at this point, we were still phenotypic. We still didn't know whether or not these compounds were really inhibiting NLRP3 specifically, and so we built a biochemical fluorescence polarization assay. And to do that, we had to sort out the problem of protein expression. So NLRP3 wants to form aggregates. It wants to come together with other NLRP3s, and it's really, really difficult to express. And so our protein sciences department did a fantastic job extracting the NAC domain of the protein, working out exactly what domain we should use. And really one of the keys was that they used one of the compounds to help to purify the compound so that the protein folded nicely as it was being purified, and it could be extracted as a monomer. We then created a probe whereby we used this compound here, so NP3146, it's just a slightly more stable version of CRID, and we attached a fluorophore to it, and then we could add this compound, this NP3301, this fluorescent compound to the protein, and then we could displace it with increasing concentrations of a molecule. In this case, we're using NP3146 itself, so we're basically displacing like for like. And in this way, we could determine which of the hits from our HTS actually inhibited NLRP3 or in the NAC domain, and which we still weren't sure about. At the time, this was absolutely revolutionary for the science. So 3 of the hit series that we had are shown here. Compound 10, this tricycle series, was a nicely potent compound that didn't inhibit in TNF alpha and was nicely active in our FP assay. And we worked on it a little bit in the early years, and we published some of that work last year. And if you wish, you can read the paper from Uri and the team, which is referenced here. We also had some other compounds like 10 and 11. You can see some structural similarities between them. And we thought that these were quite interesting, that more than one came up. And so we also decided, well, let's also work on 11 and 12. So one thing that we haven't really touched on yet in the talk is the idea to penetrate into the central nervous system. So here I show you a bunch of compounds all based on CRID, which have gone into the clinic, and none of them really have ideal properties for going into the CNS, with the exception of the compound from Novartis, which is a prodrug of an acid, because they have a lot of polarity, particularly in that core, and they have a lot of hydrogen bond donors. And so, if you were to look up the rules, if you like, of how to make a compound penetrate into the brain, you certainly wouldn't find lots of hydrogen bond donors and lots of polarity as a guideline. And so these compounds are really not ideal for a CNS application. So one of our pillars was also to look for compounds which would potentially get into the CNS. And so, we started to look at these compounds and we said, well, the phenol is exactly the same. And then you've got a ring system attached to it, which either has, which has two hydrogen bond acceptors, either in one ring or in two rings. And so maybe this black TMP motif over on compound 11 is not so needed. So what about if we sort of like do a bit of a hybrid molecule on this and put together what we can see? Because, the molecule 11 seemed to touch TNF alpha a little bit, and we were a little concerned about that. So, we made a hybrid compound 14. And what we found is that this compound, where we chopped off the TMP motif, was actually more potent than either of the starting points. So now we have a compound which is about 400 nanomolar in our cellular assay, doesn't hit TNF alpha at all, nicely potent in our FP assay and also in whole blood. It's a little bit lipophilic, a log D of 2.8, but we thought this was a really exciting starting point for our MedChem program. We were working on several chemical series. I've shown you the tricycles, compound 14, and we also had some other compounds on the go. And we started to notice similarities and commonalities in our series. What we needed tended to be like a lipophilic tail. If we added a lot of polarity, everything went south. And then we needed a central motif which had hydrogen bond acceptor character, either 1 or 2. And then there was a head group, which could be various things. And we thought, well, also if you do an overlay of the ligands, you can see here that, that they have some similarity in their overall shape and form. And so we thought, well, what about if we do another version where we do some SAR transfer? And so we decided to move the piperidine onto compound 14 and make compound 15. And that was really like a wow moment for us in the program. You can see that there's a lot of zeros after the decimal point. So we've now gone to a compound which is sub-nanomolar and which is single-digit nanomolar in our whole blood assay. This was really revolutionary at the time. Nobody had ever shown that. We were still dealing with IL-1 beta inhibitors at several hundred nanomolar in human whole blood. And so this was really a huge advance. The compound's rather polar with a log D of 1.4. But it had excellent properties. It was very stable in microsomes of mouse and human. It was very permeable, and very interestingly, we profiled it a bit more broadly. So it didn't seem to be an efflux substrate in an MDR1 cell line. So for those of you that are not familiar with this, this is a way to measure whether or not your compound may permeate into the brain. And you have a system whereby you have a membrane with tight junctions, which mirrors the blood-brain barrier, and you also have PGP expressed in it. So PGP is the protein that's expressed by the MDR1 gene, and it's the major transporter of compounds out of the brain. So the brain doesn't want to have xenobiotics in it, obviously, and so they have various transporters which remove compounds from the brain, and PGP is the major one. And so you can measure the diffusion of compounds back and forward across this membrane. In an equilibrium system, you would have the same movement from apical to basal or from basal to apical. But if your compound is a transport substrate, then it will be actively transported from the basal compartment back to the apical, and you will end up with an efflux ratio, which is the measure of the apical to the basal over the basal to the apical of above 1. And within error of the assay, this is pretty good, and shows that this compound is not a PGP substrate. So this was pretty exciting, and we wanted to move forward with this compound. I mean, at this point we had no crystal structures. It was really just ligand based. So compound 15, how important is the phenol? So we capped it or we removed it, and we found that you lost basically all your potency. And you also lost a lot of you also increased your efflux ratio. So that showed us that this viterion was very important for the overall balance of the properties of the molecule. And then we also found out that if you added a little bit more lipophilicity, so a trifluoromethyl group in this para position, you kept all the good properties of the molecules. You just raised the log D a tiny little bit, which is probably helpful in CNS drug discovery, and you also kept a very nice efflux ratio for molecule 17. We also tried to completely replace the phenol. So typical replacements for phenols can be indoles. You can see in the diagram in the top that typically you try to put the indole in where the NH replaces the phenol in an exact fashion. And this wouldn't really work here because of the steric hindrance with the protein, and there is a clash. But actually, if you look at it differently and you try to basically attack the hydrogen bond interaction with the other, in the other sense, trying to hydrogen bond with the other lone pair of the carbonyl, then that looked a lot better. So a different format for the indole. And we did that, made compound 18, where we kept pretty much all of the potency. We lost a little bit, but it was still a really nice molecule with a high lipophilic efficiency. But what we did start to buy in were problems which are associated with more lipophilic amines, including activity on the hERG channel. And that's really, although these between 17 and 18, you're not increasing your log D, you are changing the distribution of the polarity, and the zwitterionic character of 17 is really important to just balance out your properties. And it means that although 17 has a little bit of a hERG flag, we could find many compounds which were much cleaner. If you add a nitrogen to the indole and make it an AZA indole, you actually come with 19, where you increase potency and you balance the polarity a little bit, and you remove much of the hERG flag to form this molecule MP3742, which we published just a few months ago. So I think I'll skip this one actually. So we moved on, and you know, we said, well, what about the amine motif? And what we found there was that the interaction was really specific. So you know, changing the NH of the linker to an oxygen, inverting the stereochemistry, or changing the ring size all dramatically impacted the potency. And then you could remove the N-methyl. You kept the potency, but obviously you add an extra hydrogen bond donor, so you impact your efflux ratio. And then also we discovered the one thing that worked quite well was to increase the size of the substrate, just of the substituent on nitrogen, just a little bit. And there you kept all the good properties of the potency, the metabolic stability, and also the efflux ratio. And so this molecule 24, we also liked a lot. So here is a head-to-head comparison of 3 of the best molecules, which I've just shown you in the peritonitis study. So this is a bit more of a high bar study where we trigger with LPS and also ATP. It's a much harsher model. And here you can see that all 3 compounds, 15, 17, and 24, inhibit the peritonitis at 3 milligrams per kilogram, which is a really very low dose. You can also see, so 15, the blood levels and the brain levels are relatively low, but for 17 and 24, we have actually very high levels of compound in both compartments at 3 milligrams per kilogram, and we have good KPU and also an acceptable KPUU for such a tool compound. So we christened compound 24 MP3253, and we have released it to the community as a tool compound, so that people can build a data set with this compound, and that we can really build evidence for NLRP3 in different diseases, and in particularly CNS diseases. So how does this molecule bind to NLRP3? So here we can see that the lipophilic part binds in the back pocket. The phenol is the key interaction to the alanine. The arginine interaction is mediated by the pyridazine. And now we have a very strong interaction between this basic amine and the glutamate 629. It sits in exactly the same binding pocket as DFV. It just takes a slightly different path through the protein, but the interactions are the same. And so in conclusion, I've showed you that we undertook a phenotypic HTS to discover several new inhibitors of NLRP3. This has culminated in the release of this molecule NP3253 to the community as a brain-penetrant tool compound, which can be used for in vivo exploration to validate the target further, and is also useful for further optimization. And really, the pyridazines are an extremely promising compound class, which makes them of high interest for further development towards a clinical candidate. So all that remains is to thank everybody that's been involved in this project. It's been a huge effort over several years at Novartis, and then also to acknowledge my colleagues from IFM who did all the work on DFV, particularly Dongming Shen, who led the team that discovered DFV, and to Bill Rausch, who was the head of chemistry at the company. So thank you very much for your time, and I hope that we still have some time for some questions.
Awesome.
Thank you.
Yeah, thank you so much, Angela. That was a very, very nice talk, and yeah, we have a lot of applause here from the audience virtually, as you can see.
Hi.
And yeah, really very insightful story for DFV and the tool compound you showed, the brain penetrant tool compound. Really amazing how the potency ended up being so high, and you got essentially this brain penetration with very small changes. I think what you've shown us throughout the talk, very small changes can lead to significant impact on potency and other properties. So really, really amazing. We do have a lot of questions from the audience, and we have, maybe 10 or so more minutes here to talk about those. So I want to maybe quickly summarize those. One of the themes that people are wondering about is, so maybe we'll start with the first question here. What are your thoughts on pros and cons about targeting the IL-1 beta NLRP3 pathways via degradation of NEK7? So people are wondering, how do NLRP3 inhibitors compare to NEK7 degraders?
Yeah, so I think, ultimately everything will be shown in the clinic, and some people have taken NEK7 degraders into the clinic. I think, so Novartis, we published our work maybe last year, and we showed that you don't bottom out the IL-1 beta response. So there's a residual amount of IL-1 beta left. It doesn't respond as well. I think in our paper we use NP3-253 as a comparator, and so you can see the difference. And I think, is that important? We don't know, but I think that I think that's the beauty of having different targets, is that you have more shots on goal, and you should eventually have clinical efficacy. But these things really can only play out in a clinical setting.
Yeah, makes total sense. And then I think we've mentioned the elephant in the room, right? The hexahydroindazine. Was there any SAR development? Yeah, it has like the elephant ears, right?
Yeah, Mickey Mouse ears, yeah.
Or mouse ears, exactly. Has there been any development of SAR on that part, especially since potentially the aniline, could be responsible for any like toxic effects?
Yeah. So, yeah, if you were to have a look into the patent literature, maybe the Drug Hunter site could help you with that. You would find that there have been quite a lot of attempts to rework the aniline by IFM and others. I think if you can't get rid of so we have shown that aniline is not Ames positive. And so if that aniline was Ames positive, that would obviously be a different story, but we've shown that aniline is not Ames positive. And so I think by stabilizing the core, you no longer have this very, very labile core, which liberates a high amount of aniline. And so I think that we felt, or IFM felt, that it was not necessary to completely change it. And it so happens that keeping it retains many good properties. You start to run into more PK issues when you start to break it up and change it. You can get very potent compounds by changing it, but other properties start to suffer, as is always the case. And so I think, by stabilizing the core and checking that it wasn't Ames positive, I think that really, really meant that they thought that they could proceed.
Awesome. And yeah, people are wondering, did you use any other assays preclinically to rule out like DILI risks with the compound, or pretty much just went with the Ames test that you just
So IFM did run, you can run like HepG2 assays, or various versions of that. So yes, they had run, a HepG2 assay, but I think if I remember correctly, they didn't do anything in particular, a HepG2 assay, and then try a tox study. They went into tox studies. You do rat first, see how the rat looks. You don't run everything at the same time, so you can go stepwise. And then the thing to remember, of course, is that CRID3 went into humans. Now, I don't know, I don't have the Pfizer tox package, but obviously it didn't show so much tox in preclinical species that it precluded the advancement into humans, because, you know, obviously Pfizer would not have done that if it had shown very clear preclinical tox in rats or dogs. So yeah, you know, that was one of the caveats, that you weren't really expecting to see it, I think, in preclinical species, because, you know, probably they hadn't seen very much of it with the original molecule.
Yeah, that makes total sense. And there's another question in the audience. Do you consider general immunosuppression a significant safety concern for NLRP3 inhibitors in general, like a on-target immunosuppression?
No, not really. I think, you know, it's only for sterile danger signals. So you would still have the bacterial response, and to external pathogens. You know, perhaps, and we don't really see it with the IL-1 beta inhibitor, which obviously there is all pathways. You're only inhibiting IL-1 beta, but we don't have a significant infection risk with canakinumab. You know, perhaps with if somebody was ever able to bring a caspase-1 inhibitor, you know, that would inhibit all inflammasomes, and so potentially that might have a higher infection risk, but we're not overly concerned about it because it's specifically just sterile danger signals.
Yeah, makes sense. Yeah, and then Jonathan Bale is wondering about Vertex have historically been interested in caspase-1 inhibitors. Caspase-1 is activated by NLRP3, as you have shown. Do modern NLRP3 inhibitors supersede all caspase-1 inhibitors and remove the market potential for caspase-1 inhibitors?
Probably not. There will be differences. You know, I think they might do, but then because caspase-1, then you're going to be inhibiting all the inflammasomes. It's really the master switch. So I think that the NLRP3 is a more subtle approach to this problem. But who knows? Let's see. You know, I mean, really, this is, you know, you now have to run the clinical trials, you know, and see what the difference is. So I think that, yeah. Of course, I would like to think that NLRP3 would supersede the caspase.
Again, everything plays out in the clinic, right?
But yeah. It's an exciting time. You know, it'll be really, really nice to see how it works out. So
great. Yeah, and then John Overington is wondering, NLRP3 seems to be getting a lot of interest as a fix-it-all target for aging. Is there a likely difference for inflammatory suppression compared to, say, COX-2-mediated suppression of inflammation? And would a COX-NLRP3 combination have a better profile, potentially?
Maybe. Yeah, I don't know. I haven't thought about a COX-2 or a COX inhibition combo too much. There is a lot of hype at the moment, and you know, every time even you open like a newspaper, you see, you know, inflammation is chronic inflammation is the root of all evil. I think it's going to require some clinical trials, and that's also why we, you know, mining the CANTOS data has been helpful, just to point you a little bit in different directions. Potentially not having combinations, because the more combinations of anti-inflammatories together that you have, the more likely that you are to start to run into risks of immunosuppression. So I would hope that we wouldn't have to have combinations of immunosuppressants, to be honest.
Yeah, makes sense. And then do different NLRP3 inhibitors, whether targeting distinct binding sites or other components of the inflammasome, show meaningful biological different profiles in biology, or do they converge in similar downstream effects? Like basically, you know, like the glue mechanism that you showed, or other binding, or again, coming back to the neck.
Yeah. Well, I mean, neck's a different protein. I mean, I think the question was about different binding sites.
Yeah, different binding sites.
Yeah. I mean, we've focused mainly on this binding site. I can't really speak so much for other potential binding sites. So I'm not too sure, to be honest. I'm not really able to answer that question.
No worries. And have you been thinking about a PROTAC approach that Joshua Clark is wondering that could be, kind of the next approach? What do you think about a PROTAC and NLRP3?
Yeah, I'm not sure if you need to degrade NLRP3. I mean, we've shown that you can get pretty much complete inhibition of IL-1 beta. So why would we need to remove the protein and degrade it? There could be reasons to do that, but these molecules are very small. Each atom counts, as you said. Very small changes make big differences, and I like the small molecules. I wouldn't go for a PROTAC unless there was really a reason to do that in terms of some scaffolding function of NLRP3, or that you have to really remove all the action of NLRP3 to really have the effect. So unless we need to get down to 100% inhibition, and 99% won't do, I'm not totally sure why we would need a PROTAC, really.
Yeah, makes sense. Well, we have a lot more questions, but unfortunately we kind of ran out of time. So sorry about that. But yeah, we will probably get back to you on some of those questions. So let's thank Angela again for a wonderful talk about NLRP3 and drugging NLRP3, the NLRP3 inflammasome. So really very insightful talk with a lot of learnings and lessons that can be taken away by the audience. Thank you again, Angela. Really appreciate it. And sticking around for the audience questions as well.
No problem. Thanks very much for the opportunity, Dennis. And
Absolutely.
Good evening to everybody. Thanks for listening in.
Yeah, thank you to the audience as well for the engagement and all the questions. We really appreciate that. And we will be back here next week with a flash talk. That'll be a year-end special. That'll be actually a flash talk by myself about the 2024 molecules of the years and the scientific highlights of those. So yeah, that'll be an interesting flash talk to dial into. And with that, I wish everybody a wonderful rest of their week. And thank you again, Angela. Thanks to the audience. And we will see you here next week. Take care.
Bye, everybody.
Bye-bye.